Hello all, welcome back to the Biorocks channel. From today, I'll be releasing few of the videos every week on the genetics series. This whole series and series of these videos is completely dedicated to the genetics. I hope these videos will be helping you all in understanding genetics better. Okay, in this first video today, what I'll be doing is to make it very clear and to make it very understandable and easy accessible about the terminologies and about the concepts of the genetics. I'm starting it with the chromosomal structure. So today's topic is going to be about the chromosomal structure. Okay, so when I speak about the chromosomal structure, that means that I'm speaking about the tertiary structure of the DNA. This is nothing but I'm talking about the tertiary structure of DNA. I have already released one of the videos in the Biorocks regarding the nucleotides, that is nucleic acids, in very first few of the videos that I have released. So you can just go on to that particular video to just find out the more information about the nucleic acids, where I spoke complete details about the DNA. So in this video, as I'm speaking about the genetics, I'm starting with the chromosomal structure, which means that I'm talking about the tertiary structure of the DNA. That is about the how this particular DNA molecule, which is very, very complex, very longer complex structure, which is getting packed up, okay, into a structure which is being called as a chromosome. So here in this video, I'm talking about the chromosomal structure, which means the tertiary packing of the DNA, structure of the DNA, and I'll be talking in detail about the chromosomal structure. So first, talking about children, before I start my video, I have two of the questions that have to be raised before I enter into the this particular topic. The first thing is, like, when we speak about the length of the genetic material, the length of the genetic material is far exceeding the length of your cell. Okay, but the cell has to actually come up with a mechanism where it can actually pack this a longer and a lengthier genetic material into such a small volume of cell. So your cells have to come up with this mechanism. So we will find out that what is that mechanism that the longer, lengthier genetic materials, which are far exceeding the length of the cell, how come are they getting packed up inside the cell, as well as the second thing is about how they are being folded, how they are being packed up very tightly. Okay, and the second thing is like the genetic material, in order to get replicated or in order to get transcribed, they need to be actually unfolded from this tightly packed structure. You are able to understand the two questions that I have raised. Is the first thing is, the first question that I am raising is your genetic material length is going to length far exceeds the excuse me, far exceeds the length of the cell, right? So what we need to do is, here we need to tightly pack it, right? We need to tightly pack the genetic material. Okay, this will be the one, the material. One thing is, I spoke that how can we actually place this particular lengthier genetic material into a small cell, and the answer for this was to tightly pack the genetic material. But the second thing is that if it is being tightly packed, if it's tightly packed, then how is that the genetic material is getting transcribed, and how is it getting accessible? It's getting accessible to the enzymes, that are playing a role in replication of your DNA. right? See, once if it's been tightly packed, if it's been tightly packed, how can it be accessible for transcription? That is, there are many factors which are going to actually help in transcription. So how can this DNA be accessible to the transcriptional factors? How can it be accessible to the enzymes that are helping in the replication process? So one thing here is very clear, children, that in order to transcribe and in order to replicate, your DNA, which is tightly packed, has to be actually unfolded, right? One thing is we are folding it very tightly, and this has to be unfolded, and back again it has to be folded. So this need to be enlightened, or this need to be highlighted, because these should be the features. Now, one thing is we told that we need to tightly pack them. If they are packing, we can't transcribe, we can't replicate it, but in order to make it accessible, I need to come up with a concept of folding and unfolding. So who does this folding and unfolding? This folding and unfolding is being catalyzed by one of the enzymes, which is being called as Topo Isomerase. So this is an enzyme which is going to fold and unfold the DNA based on the time when it has to be transcribed and replicated. And once if it's been done with, it has to be folded back again. So that's being done by the enzyme which is being called as a Topo Isomerase. Okay, children. So now I'm entering into the topic where I've started up with the length of the genetic material exceeding the length of the cell. I told you that it has to be tightly packed. So how are they being tightly packed? So here what I'll try to do is, let me speak about that your DNA is being actually super coiled. Okay, your DNA is being super coiled. DNA is being super coiled. So what is this DNA super coiling? Is nothing but it gets highly coiled up, highly folded up, such that it can occupy very little space inside the nucleus. Okay, so because we need to accommodate the space for this DNA inside the nuclei, which is very lesser than that of your cell. So we are super coiling, super folding your DNA. And when we speak about this supercoiling, I told you that the topoisomerase is one of the enzymes which is going to help in folding and unfolding. There are two types of these supercoilings. One is being called as a positive supercoiling, and the other is being called as a negative supercoiling. Okay, usually your DNA is going to be in a state of relaxed state. For this, let me share my screen with you all, children, such that I can give you this concept well about the coiling process. Okay, so if this particular thing that you're seeing in the center is going to be the relaxed DNA. Okay, that is going to be your relaxed DNA. Let me take my marker such that I can explain you better. So this one is going to be a relaxed DNA. If we speak about the relaxed DNA, that is a bDNA, usually every turn is going to have some 10 base pairs, okay? But here you are seeing the positive supercoiling. This positive supercoiling is nothing but, if you have the older versions of the telephones, you can see that there is a long wire, the coiled wire. So in the same way, if such a coiled wire is being tightly coiled up, very folded up and tightly coiled up, it is going to increase the number of coils, right? So here in the super relaxed DNA, there are 10 base pairs per one turn. Okay, but if it is going to be positively coiled, what happens is the number of turns you have increased, right? You have increased the number of turns. But if it is going to be negatively supercoiling, what happens is the loops are very loosely coiled. The loops are, the terms are very loosely coiled here. Loosely coiled. These are very tightly coiled, and these are going to actually fold above itself, and it's going to be over rotated. Now let me share my screen with you all and show you actually. Let me remove this. Yeah, so I'm talking about the supercoiling of your DNA. One is positively supercoiled and one is negatively supercoiled. So children, for instance, if I'll take an easy diagram such that I can explain you. So for instance, if this is your DNA coiled up and this is your one of the turns where it is accommodating 10 base pairs, and this is the relaxed state of your DNA. Okay, but if I'm going to take the one which is going to be the tightly coiled, which is a positively coiled, okay, this is your positive supercoil structure, where you can see that the number of turns have got increased and the accommodation of the base pairs has reduced a lot, because here you can see that it's like 1 2 3 4 5 6 7 8, and you can see that the number of coils have the number of base pairs for one turn have reduced a lot because of this positive supercoiling, right? Whereas in the relaxed state, one turn is accommodating 10 base pairs. In the same way, if I'm going to take one of the negatively coiled, this is negatively supercoiled, where you have reduced the number of turns. The number of turns are going to get completely reduced, and now what happens is the number of accommodation of the base pairs is going to usually increase in this. It's not like that. It's going to be the number of turns have reduced, but the base pairs are going to be more for this. And if you're going to look at the number of turns, it's going to be only like you can speak about that. For example, let me give you one particular comparison where I can speak about, for instance, if I'm going to have 100 base pairs in the relaxed state, I'll be having around like 10 turns, right? I think now you have understood this. So if I have 100 base pairs, usually have 10 turns to accommodate these 100 base pairs. But if I'm going to talk about the negative coil, where it is loosely coiled, okay, loosely coiled ones, here you can see that what happens is the number of turns are going to be very less. It might be only 9 to 8, because the number of base pair accommodation here is high. So usually what happens, like we can have only 9 of them, not the 10 of them to accommodate them, okay? And you can see, because the number of the base pair accommodation has increased within one turn, okay, this is very important. But when compared to that of the positive ones, what happens is the number of turns have increased and the accommodation of the base pairs have reduced. So children, you can think about, here you can have 11 to 12 turns in order to accommodate the same number of 100 base pairs here. But here what has happened is, since the accommodation of the base pairs are very higher here, we have seen that the number of turns have become very less here. This is the concept of the supercoiling. I think now you are aware that, as I told, that the genetic material far exceeds the length of the cell. It needs to be tightly packed, and in order to tightly pack, we have a concept which is being called as a supercoiling, where you can have either the positive coiling or the negative coiling. But children, here an important thing to note is most of the DNA in our cells are negatively coiled. Let me write this for you, okay? So let me write it as, most of the DNA in the cells are negatively coiled. Okay, why are they negatively coiled, children? Because I told you that here it's loosely coiled, and you know better that in order to have a good transcription and replication, your DNA has to be unfolded, or it has to be loosely packed or loosely coiled. So now for this concept, what we are doing is, we are coming to discover that most of the DNA in the cells is negatively coiled, because in order to have a rapid separation, very quick unfolding or separation during the replication process, and secondly, it even requires very little energy to unfold the negatively coiled DNA. Are you understanding it? Because here the amount of the turns are less, so you require very less energy. Our investment of the energy for the unfolding is going to be very less when compared to that of the positively coiled DNA. I hope this is clear for you. The concept is clear, children. Now coming on to, yeah, the next thing is that I have to speak about here is, the first thing is about the DNA of your bacteria. Let me first start with your prokaryotes. Let me share my screen with you all such that I can give you a glimpse of the bacteria. Yeah, children, that you're seeing on the screen is the bacteria. If you are going to remove the DNA of, if you're going to remove the DNA of the bacteria, that is a prokaryote, you can see that it's going to be like you can see some loop-like structures, right? These are the loops that you can see in the chromosome or the DNA of your bacteria. Children, if I'm going to speak about the E. coli bacteria, that is going to have a 4.5 to 4.6 million base pairs. And if you're going to stretch this particular 4.5 to 4.6 million base pair DNA, this is going to be 1,000 times longer than the E. coli that you can see on the screen. And you have to understand that even the prokaryotes needed to evolve a mechanism such that they can actually pack this lengthier and a longer DNA into such a minute body. Okay, and if I'm going to speak about the packing here in the bacteria, let me share my screen with you. So usually the bacterias are going to have a single circular DNA, and this singular circular DNA is going to be highly looped structure, and this looped structure is forming a complex. DNA is forming a complex with the protein, and these proteins are basically giving that structure of loops and attachment of the DNA. Okay, so for instance, if these are going to be the proteins, they are going to give a prime foundation for forming the loop-like structures for this particular DNA, the circular DNA of the bacteria. But remember, children, the prokaryotic proteins here are not the histone proteins that we are going to find in the eukaryotic cells. Okay, and if you look at a bacterial cell, it does not have a nuclear membrane, or it does not have a nuclear envelope, nor the organelles are covered by the membrane, so we were calling them as prokaryotes. But the DNA, or this particular bacterial chromosome or the bacterial DNA, is being present in a discrete what you call is discrete place, and it is going to form a structure which is being called as a nucleoid. We are going to call it as nucleoid. Besides this, the bacteria is also going to contain a double-stranded DNA, which is very independent of this particular chromosomal DNA. Okay, it's independent of that. It can replicate on its own without the orders of this particular chromosomal DNA of the bacteria, and this structure is being called as plasmid. In the forthcoming series of the videos, when we speak about the molecular genetics, when we speak about the biotechnology, we'll be learning more about this particular plasmid. Remember that plasmid is independent of the what you call is a chromosomal DNA. This is important to remember. Okay, and when we speak about this, when we compare the prokaryotic chromosomes with that of the eukaryotic chromosomes are going to be very longer, and these are going to be the very complex structures with that of the histone proteins. Here we do not find the histone proteins. So now what I'm talking is about the eukaryotic chromosomes. eukaryotic chromosomes. So when I'm talking about the eukaryotic chromosomes, children, I'm telling you that these are going to be very long molecules of DNA with the histone proteins. Okay, these are the long molecules of DNAs, complexes with the histone proteins. And especially you can see that during the interphase and during the cell cycle, we can see that these long molecules of DNA are going to, let me write, during the interphase and during the cell cycle of mitosis and meiosis, these long molecules of DNA histone proteins, which are the chromatin material, are going to condense. Okay, so the chromatin is going to condense to form the short stout structures which are being called as chromosomes. And remember, children, why is condensation important? Why do we require this condensation? We need this condensation because the condensation is going to help in a proper movement. This is going to help in the movement of chromosomes during mitosis and meiosis. This is important point that you need to understand. So I'm trying to compare with the chromosomal DNA, the chromosomes with that of the eukaryotic chromosomes with that of the prokaryotic chromosomes, where we have seen that the prokaryotic bacterial chromosomes are forming the complexes with the non-histone, or they are the simple proteins, actually, not even the non-histone proteins. They are the simple proteins that are forming the complexes, forming a loop-like structure. While in the eukaryotic chromosomes, the long molecules of the DNA making up a complex with the histone proteins, and these long molecular DNA structures, which are the chromatin, are going to condense to form a short stout structures like chromosomes. And forming a or undergoing condensation is very, very important for us, because this is going to enable the movement of the chromosomes during the anaphase, where you can see that they split into a pair. Okay, and remember, children, the change of the chromatin, that is changes of the chromatin that is forming into chromosomes, forming condensation, undergoing condensation, is going to be a very important dynamic process. Again, it's a very dynamic process. The first thing is this, and the second thing is condensation is going to be a very dynamic process in DNA replication. And gene expression that we are going to see in the forthcoming videos of the genetics. So these are the reasons for why your chromatin is going to get condensed. Okay, and coming on to then, how do I define what is chromatin? What is chromatin? Chromatin is nothing but, this is going to be your DNA molecule with your histone protein. Okay, these are going to form the DNA, and the protein is going to form the chromatin. Okay, now talking about this chromatin, children, there are two different types of chromatin. One is being called as your Euchromatin, and the other is being called as a heterochromatin. Okay, so the Euchromatin is the one which is going to actually undergo the, what do you call, is the normal cycle of condensation and decondensation. So this is the one which is going to undergo the condensation, decondensation process during the cell cycle. Okay, and the other important thing here is, this is a one which is going to contain many number of genes, and these genes which are going to encode the proteins. Okay, so this is going to be your euchromatin. So during the cell cycle, during the that is a cell life cycle also, you can see that many number of the genes are going to encode the proteins that you will learn in the central dogma in the forthcoming videos. So just remember remember that the euchromatin is that part of the chromatin which is going to undergo actual condensation and the decondensation processes. And coming on to the heterochromatin, children, this is going to be the one which is going to just remain in a very highly condensed state. You already know, I have told you that if they're going to be tightly packed children, I have told you that can they be accessible for the transcription and the replication? No, they can't, right? So in the same way here, you can see that your heterochromatin is remaining in highly condensed state, even during your mitosis and meiosis. Even during the mitosis and meiosis, they stay in a highly condensed state. But where do you find this heterochromatin? Heterochromatin can be found in the structures of your chromosomes, especially in the telomere section. You can find them in the centromere structure. You can find them, for example, if this is going to be your chromosome, we can see that this structure is going to be the centromere, and the tips of the chromosomes are being called as telomeres. So this is centromere and this is going to be telomere. So those parts of the chromosome are going to be remaining highly condensed even during the mitosis and meiosis, and these are the ones which are going to contain the heterochromatin. And remember children here that the heterochromatin do not code for any, or that this does not encode any particular gene at all, so no protein production here. And remember that there is no transcription happening here, no transcription happening, as well as there is no crossing over that happens here, not even the crossing over happening in this particular state. Remember, there are even different types of heterochromatins, like the constructive, that is a constitutive, and the facultative. The constitutive is something nothing but the ones which are being present, the telomere and centromere, which are highly, completely, permanently condensed, which do not encode any particular gene. Okay, that is going to be your heterochromatin. I hope you understood what is chromatin. Chromatin is a DNA with the complexes of the protein, that is a histone proteins, and your chromatin is of two types based on how active it is transcribing or how active it is participating in encoding the genes, whether or not encoding the genes. Okay, now next thing I would like to say you is about the proteins. In chromatin, I was talking about the DNA is forming a complex with the protein, and we were calling it as what? We were calling this as chromatin. Now coming on to the protein. So who is this protein? The protein is nothing but these are the proteins which are going to be called as histone proteins. As I told you that your DNA is very longer, lengthier structure, has to be tightly coiled, and the coiled structure is going to be, the coiling technique may be negative or the positive, but some of them should be there to pack them, and those are nothing but the positively charged. These are positively charged proteins which are being called as the histone proteins, and you far better know that your DNA is negatively charged, and because of their opposite chargers, there is a tight attraction between both of them, and moreover your DNA is coiling and it is going to get packed up with the negatively charged, oppositely charged protein which is being called as histone proteins. And as you all know that there are different types of histone proteins. Let me share my screen with you all to show this particular process of coiling. Okay, children, let me go to the histone proteins. Yeah. So if I'm going to talk about the histone proteins, children, there are different types of histone proteins here. As you can see on this particular screen now, that there are H2A, H2B, H3, H4. These are the different types of histones which form the octamers. What is octamer? It's a number of 8. So how can they be forming 8? Is nothing but 2 of each of the H2A, 2 of the H2Bs, 2 of the H3s, two of the H4s totally form the octomer protein. Okay, and you know that your DNA is going to coil either negatively or positively. So the coiled DNA, you can see this red colored DNA which is coiling about the octomer. You can see 1, 2, 3, 4, 5, 6, 7, and 8 of the octomer histone proteins getting attracted with the negatively charged DNA here. And you can even see that there is a DNA, there is an histone protein here which is being called as H1 histone, which is going to actually seal away this particular structure. I'll come to that details also, but just I want you to remember that there are histone proteins which are trying to help in coiling the DNA because of their positive charges. And you can see these black structures hanging away out. These are also the histone tails which are positively charged. Okay, and remember children, that this octomer protein is helping in coiling away your DNA, and the DNA which is coiling over the octomer is being called as a core DNA. So a core DNA plus a octomer has to be regarded as, what do we call this as? We call this as a nucleosome. I hope this is clear for you now. Okay, now let me go to, yes, let me exit this. Yeah, so let me come and share you this particular structure. So you can see that these are your green colored ball-like structures are the histone proteins. Let me first, yeah, so let me share you my screen with you all. So I spoke that there are 8 of these histone proteins. Okay, these are forming up. So they are going to be made up of H2A, H2B, H3, and H4, right? So you can have H2As, H2Bs, 2 of them, H3s, H4s, togetherly forming a octomer. And later, about this particular octomer, we have spoke that the DNA is going to coil. It's going to be the coiling of the DNA. The DNA which is coiling on the octomer, this is your octomer, and this DNA is going to be called as core DNA. These togetherly are going to form a structure which is being called as nucleosome. Okay, this is going to form the nucleosome, and this nucleosome is going to be now sealed completely. This is going to be sealed by one of the histone protein which is being called as H1. Okay, if this particular H1 is packing it, then the, see, nucleosome with a H1 protein should be called as what? This should be called as a chromatosome. It's a chromatosome. Let me first erase this such that when you study, this will be becoming easy for you. Let me, give me a moment. So I spoke about the octomer protein with a core DNA coiling, right? So this is nucleosome. Now, what is a chromatosome? A chromatosome is nothing but the histone protein, that is octomers, with the core DNA sealed with, this packing is sealed with a H1 protein, and this is your octomer, and this is going to be your core DNA. Then we should call this structure as what? This structure is being called as a chromatosome. Got it, children? This is very, very important. Remember that even your histones were having these tails, right? These histone tails are also positively charged, and these are the ones which are going to undergo acetylation, methylation, and they will be facing up, or they are going to actually undergo the epigenetic process that we'll be seeing in the forthcoming videos. I hope now you got a clear idea of your DNA, that this is going to be super coiled. It may be negatively coiled or positively coiled, and we have compared it with the prokaryotic DNA, and we spoke about the DNA, speaking about the chromosome. That is, we spoke first of all about the chromatin. We spoke about the types of the chromatins which have been present. We spoke about the histone proteins. We spoke about the nucleosome. We spoke about the chromatosome. Okay, especially children, these histone proteins are going to contain two of the amino acids which are being called as your arginine, as well as lysine residues are many in this particular histone proteins. Okay, I think till here your concept is clear. Now coming on to how is a tertiary structure of DNA is formed. Okay, how is it being formed? What is the first step? What did I say? I told you that the DNA is going to form a complex with the protein, and this is going to be actually your chromatin. And what did I say? That the chromatin is going to have many repeats, right? So the fundamental repeats of, how is chromatin? I told you that it's the core DNA with this. What was this? The core DNA and the octamer protein, right? Then you have another octamer, and here comes another coil, and it goes away. This particular DNA, which is hanging in between two of the nucleosomes, is being called as linker DNA. Okay, what is this structure called as? This structure is called as nucleosome. And what did I say? If this is going to contain a H1 sealing, then I was calling this structure as a chromatosome. Okay, so we can see that the fundamental repeats of the nucleosomes are found in the chromatin, and this is going to be around like a 30 nanometer fiber, the chromatin material. Okay, so you can see that the chromatin is going to have this repetitive structures. Okay, the repetitive nucleosome structures are being present. This is in a diagrammatic way I'm giving you. And remember that this particular DNA, if it is going to be highly coiled, you can see that in between you can see this structure which is being called as a linker DNA. Remember children, if it's going to be a positively coiled DNA, I told you that they are completely tightly packed. Then you can see that there will be no scope of the visibility of the linker DNA, because what is the reason? Because they are completely tightly packed in the positively coiled DNA. Since it's very tightly coiled, what happens? The linker DNA is not visible. Okay, the linker DNA is not even subjectable for the enzymatic actions also, like nucleases. For example, if it is a negatively coiled DNA, and if I'm going to take up nuclease enzyme, since the linker DNA is more visible and exposable, the nucleases are going to cut the linker DNA. But this will not be the case in the tightly coiled, positively coiled DNAs. Okay, this is very important. So if I'm coming to the complete structure about the tertiary packing of the DNA, children, first thing is I have my nucleotides. Okay, these nucleotides have formed a nucleic acid which is being called as a double-stranded DNA. Right? Later, the double-stranded DNA, you saw that an octamer protein is packing up the DNA, and this is wrapping up, and this is forming a nucleosome structure. Right? These repetitive nucleosome structures, see, remember the DNA is coiling, right? How many coils? It is going to be 1.65 times it's going to coil up. Okay, and you remember that this particular octamer DNA, it's forming a nucleosome, and I told you that your nucleosome is going to actually, if it is going to have a nucleosome structure with the H1 protein, is being called as a chromatosome. Okay. Secondly, if your nucleosome is going to, and I told you that this nucleosome is going to be actually folding up, it is going to fold to a 30 nanometer fiber. Okay. And this is going to further form loop. This is going to form loops around 300 nanometer in length. Okay, first thing is a nucleosome. Nucleosome is folding into a 30 nanometer fiber. Further this is going to form the loops, and those loops will be 300 nanometer length, and later you can see that these are going to be more folding up into 300 nanometer fibers, which will be further tightly coiled up, producing tightly coiling is nothing but condensing. So now the chromatin is condensing into what? Producing the chromatids, forming the chromatids. That is of chromatids of whom? Of a chromosome. So this is the way your DNA is being packed. If you are not well versed with the nucleotides and the nucleic acids, I am begging you to just get into the Biorocks channel and please search for that particular video that I have made about the nucleic acids. It's one of the most informative videos that you have on YouTube. Okay, just coming on to children, wrapping this up with the images. I would like to share images with you such that now the topic will be easy for you before I wrap up this particular thing. Okay, give me a moment. Just coming on. One second. Yeah. Would like to share the screen now. Yeah. First, I was talking about the DNA super coiling that you can see here, the relaxed DNA, the positive and the negative super coils, which are trying to coil up in order to reduce the volume that it's going to occupy. Now coming on to the nucleosome, where you can see the yellow colored structures, which are going to be these yellow colored structures that you can see are the octomers, where the DNA is wrapping up for the 1.65 turns, making a core DNA. And you can see that this in between, you can see that this is going to be a linker DNA, which is subjected to the enzymatic actions in the negatively coiled DNA. And next, coming on to the coming up with the next structure, which is going to be a chromatosome, where you can see that you have the octomer proteins with the linker DNA, with the core DNA. This is going to be your linker DNA, this is your core DNA, and you can see that this is going to be the histone 1, the gray colored structure, which is going to be forming the whole, that is with the octomer, the histone 1, and with that of the octomers and the core DNA is going to be forming the chromatosome structure. Now coming on to the DNA structure, let me share you this one. Yeah, you can see that the DNA is having the nucleosome. Children, let me show you this. Please give me a moment for this. Oh my, yes. One sec. I'm sharing my screen with you. So talking about this particular one. The nucleosome that you can see is Euchromatin because they are loosely packed, right? And the linker DNA is subjective for the transcription as well as a replication. You can see that in the heterochromatin, in this particular section, you can see that they are tightly packed and there is no visibility of the linker DNA which can undergo the enzymatic actions. Okay, so now you can see that this is your histone protein, which is forming the octomer proteins with the tails, and these tails are also positively charged and subjected to what? They are also subjected to the what you call is epigenetics, the enzymatic actions and the epigenetics. Okay, coming on to the DNA packing, let me show you this structure, how the DNA is being packed up. So if you look at this double helical structure of the DNA, it's forming the nucleosome structures, and these nucleosome structures are forming for the chromatin fiber, which is going to be highly condensed together to form a structure which is being called as chromosomes, as I have shown you and explained you in this particular video. Okay, children, I hope this video has made it clear for you about the euchromatin, heterochromatin, and what is linker DNA, how do they undergo the transcription and replication, and what is coiling of the DNA. And moreover, I think you have understood the concept of these enzymatic actions and how the DNA has to be loosely coiled in the form of euchromatin, which is making the genetic material to be passed down to the offsprings and to the different generations together without any particular change, but they're subject to because they are going to undergo even the crossing over and the replications, and most over the most genetic fundamentals which are very, very important during the sexual reproduction, where you can see the new features in the offsprings. These all are just because of the euchromatin and the linker DNA is present there, but not in the heterochromatin, which are very, very tightly packed, not subjected to the enzymatic actions. I hope this video has helped you all. Please do comment in the comment section how you have liked this particular video and how this has helped you all in understanding. Thank you all. Thank you.